Hello AP Biology students, this is Mr. Laudermouth. Good morning, good afternoon, or good evening to you as the case may be. This screencast is part 2 of Cells and Cell Organelles Doing Life's Work. So in the previous video we mentioned that cells have 3 basic jobs that they have to accomplish, one of which was producing energy for the cell or for the entire organism. Let's look at job number 2. Cells need to produce proteins, which basically do all the work of the cell. Cells have to read their genetic material. They have to use that DNA to then inform the building of proteins, which could be structural, which could be enzymes for speeding up chemical reactions, could be signaling molecules like hormones or cell membrane receptors. Organelles involved in the work of producing proteins include the nucleus, ribosomes, endoplasmic reticulum or ER, Golgi apparatus. How are we going to produce these proteins? Well, first we have to start with the genes, the sequence of base pairs in the DNA. That will give us the instructions we need to actually build a functional protein. Now that process of reading the genetic code we'll cover in greater detail in a future unit. But for right now, it's enough to understand that DNA contains the instructions for making the protein, and the proteins then do the work necessary to keep cells alive. Structural, enzymes, signals, receptors, etc. At the control center of the cell, we'll find the nucleus, where the DNA is housed and protected. Surrounding the nucleus, we have a nuclear membrane. Within the nucleus is a smaller region called the nucleolus. This is where the ribosomes are assembled. And of course, we'll find DNA in the form of chromosomes, which is actually the DNA double helix wrapped around proteins. The ribosomes we can think of as being protein factories. They are the sites where the genetic information, the genetic code, is translated into a sequence of amino acids. It's where that primary protein structure gets assembled. There are some ribosomes that float freely through the cytoplasm, but there are also some that are attached to the endoplasmic reticulum. They give it a bumpy appearance when you look at it in the microscope, so we call it the rough ER. So the ER is a major site for protein synthesis. The rough ER, again, contains ribosomes whose job it is to make polypeptides. The ER is also a place where proteins get folded properly, where they can be sorted, and where they can be sent to different spots in the cell. The ER is also a place where membranes and other lipids can be assembled. Rough ER has ribosomes, works on proteins. The ER that lacks ribosomes is called smooth because it does not have a bumpy appearance when you look at it in the microscope. That's mainly a site for lipid synthesis, a place where fats or the membranes that are constructed from the fats are put together. Then we get to the Golgi apparatus or the Golgi body. This is sort of the finishing center, the packaging center of the cell. It takes the products that were made at the ER, it gives them a correct shape, it can assemble, it can put together different pieces, it can label them with tags, essentially little protein molecules that direct them to different parts of the cell, or it can even prepare molecules to be shipped out of the cell for use somewhere else. Think of it like the UPS headquarters of the cell. It receives and it ships. How is it transporting things around? Well, the same way we talked about in the other video, using those little membrane-bound sacs called vesicles. They surround the proteins or other molecules that are brought to the Golgi, and they are sent from the Golgi in vesicles as well. Think of the vesicles as UPS trucks. So here, let's do a quick kind of overview of the process I just described. We start in the nucleus, where we'll find the DNA containing the instructions for building a particular protein. That DNA is transcribed into RNA, another process we'll look at more closely in the future. The RNA leaves the nucleus heads over to the ER, where it will encounter ribosomes. That RNA is translated, it's basically read at the ribosome, and used to put a chain of amino acids together in the correct order, used to build the primary structure of a polypeptide. That protein makes its way through the ER until it's packaged up inside a vesicle. That vesicle travels a ways across the cytoplasm until it reaches the Golgi. It then enters the Golgi apparatus, where it is folded correctly. Once it's in its final state, ready for use, it's repackaged by the Golgi into a new vesicle and can be sent to its final destination, which might be inside the same cell, or it might be some other part of the body, in which case it has to leave the cell. It'll exit through the cell membrane and be sent, for instance, through the blood to a different destination. Final job all cells need to accomplish: they have to reproduce. They have to make more cells. We need cells to replace ones that become damaged through use or disease. We have to repair injury, and of course there's just the simple process of growth and development in an organism. To correctly produce more cells, we have to copy DNA, have to make extra organelles. Any daughter cells produced will need mitochondria and ER and ribosomes and Golgi and all the other parts we've been discussing. Those organelles and DNA need to be divided up evenly between the two daughter cells. That process, you might recall from your first year in biology, it involves mitosis, the division of the nucleus, as well as cytokinesis, division of the cytoplasm. And, I can say it again, we'll look at those processes a lot more closely in a future unit. But for right now, let's focus on the cell parts that are doing most of this work, the nucleus and the centrioles. Centrioles are organelles that come in pairs and they help coordinate cell division. We only find them in animal cells. Plant cells, of course, have to accomplish the same job. They just use a slightly different mechanism. You'll find one pair of centrioles at each end of a dividing cell. They help produce the spindle apparatus, the fibers that attach to the chromosomes, and then pull them apart. Finally, let's look at the cytoskeleton. When I say cytoskeleton, I'm basically referring to an internal set of tubes or fibers that help give the cell an internal support structure, kind of a framework, a scaffolding inside the cell. The largest tubes you'll find are called microtubules. They can actually form something of a highway inside the cell. Special molecules can actually walk along the microtubules, pulling organelles with them. Cells actually can move organelles around inside the cytoplasm using this microtubule highway. There are also fibers called actin filaments. They form a very dense network that help to keep the animal cell's shape intact. They are also part of muscle fibers because they can contract, they can pull against each other. You'll find intermediate filaments. These help connect different cells together to form tissue layers, and they help strengthen cells. And as I mentioned, there are also motor molecules. There are proteins that can attach and detach to these cytoskeleton fibers and actually sort of walk along them, pulling organelles with them, basically a transport system within the cell cytoplasm. So to summarize, cells have to make energy. That process requires food and oxygen. It's a process of cell respiration in both plant and animal cells, and in plant cells, which possess chloroplasts, they can also make their food during photosynthesis. They have to remove their wastes that are generated. All cells need to make proteins by reading the instructions in their DNA. Those instructions can then be sent to a ribosome. The protein can then be transported through the ER to the Golgi, where it is finished and then packaged and sent on its way. Finally, Cells have to make more cells. This requires copying of DNA and dividing up the organelles, the cytoplasm, and the DNA among the daughter cells. These jobs are also important. We will study them in greater detail. For right now, it's okay just to have an overview of the parts of the cell involved in these important jobs. That's the end of our cellular story. If you have any questions, be sure to ask in class. I'll see you then.